friends, now we are going to see about the differences between histone as well as non-histone proteins. What are the proteins will comes under histone and what are the proteins will comes under non-histone. So these histone proteins are present inside the chromatin material. What exactly the chromatin material? The chromatin material is nothing but a colored fibrous seen through the microscope inside the nucleus. So inside this fibers we have the DNA. Along with the DNA is being bounded with histone proteins. So DNA, histone, together we call it as chromatin fibers. Along with this we have one more extra proteins which we call it as non-histone proteins. In order to know the difference between this, so let's see into the topic. The difference between histone and non-histone proteins are simple. Both are proteins. Both provide the structure to DNA. Both are components of the chromatin only. So chromatin is not simply with the histone. Both histone, non-histone and DNA. All the three together we call it as chromatin material. Their chief difference is in the structure they provide. So histone proteins are the spools about which the DNA winds, whereas the non-histone proteins provide a scaffolding structure. Scaffolding means for unwinding structure. Another way to think of the difference is that non-histone proteins are those proteins remaining after all the histone proteins has been removed from the chromatin. For example, we have a several meters of DNA. The whole several meters of the DNA has to compacted into that small nucleus. So in order to compact in that small nucleus, we need a adhering factor. That adhering factor is the histone proteins. For example, DNA is negatively charged. For that negatively charged, we need a positively charged particle, that is nothing but histone proteins. So the DNA wrap around the histone proteins. The negatively charged DNA will wrap around the positively charged histone proteins. And we have one more another proteins which we call it as non-histone proteins, which is in turn negatively charged same as DNA, because sometimes for replication and transcription the DNA has to be unwinded. At that time non-histone proteins will repel the DNA, negative negative will repel and the DNA will unwind and that will follow the replication. So that is the use of major use of histone and non-histone. For histone is for binding tightly, non-histone is for releasing the DNA from the binding, but all the three is accommodated together to form a chromatin material. Now chromatin. Histone are the principal proteins component of the chromatin. Chromatin is a complex of nucleic acid, DNA or RNA, and proteins which is histones, which condenses to form a chromosome during cell division. Think of the chromatin as a vacuum packing that allows a greater volume of clothes to be fit neatly into the drawer. Without chromatin, a single cell worth of DNA would unwind to a unwieldy length of 1.8 meters. On the top of packing, chromatin strengthens the DNA so that during cell division in either mitosis or meiosis, the DNA doesn't lose the structural integrity because of this chromatin material. If only DNA is there inside the nucleus, because DNA is essential for the blueprint of our body. If the DNA has to transform from generations to generation, it can cut easily in between. So it will lose that structural integrity. In order to maintain the structural integrity, it has to be tightly packed with histone proteins. So this is the picture of the chromosomes. This word chromosome can only be found during the cell division, because chromosome is nothing but a tightly packed condensed chromatin material, we call it as chromosome. So this is centrosome. So here we have the centromere. So this is the centromere. So nothing but the center part of the chromosome. And this is the chromatin fiber microscopic picture. So in this we have many beads. So this beads we call it as nucleosome. So here we can able to see the histone proteins as well as the DNA. This blue color part is the DNA and here we have the histone proteins. And here, so after unwinding this, we will get like this appearance, bead and thread appearance. In this bead and thread, the thread is nothing but the DNA. The bead is nothing but nucleosome in which we octamer. So totally we have 5 histone proteins. What are the 5 histone proteins means? H1, H2A, H2B, H3 and H4. The H1 protein, H histone protein will lines between the thread and the bead. So that histone protein we call it as linker histone protein. And next, H2A, H2B, H3 and H4, which will form a octamer or a core of this nucleosome. The center is being formed by this four proteins with the multiples of 2. So because of that we call it as octamer, means 8 histone proteins will be there with the multiples of 2 each. So bead on string, which will form the chromatin material. And this is the DNA, double stranded helical structure is the DNA. Histone proteins are the proteins that provide an essential structure to DNA, making the life possible. The protein functions as spools about which the DNA winds, a single cell's worth of unwound human chromosomal DNA, for example, would stretch around 1.8 meter. So, thanks to the histone proteins, the wound up compressed DNA takes up only about 90 millimeters of space. For this 1.8 meters of DNA is being winded into 90 millimeters of space. So that is all possible because of histone proteins. So always we have to remind the DNA is negatively charged. The histones are positively charged. Again, in turn, non-histone proteins are negatively charged. So histone proteins is used for the DNA for packing. Non-histone proteins is used for the DNA for unpacking or unwinding during transcription, replication, during these processes. Without histone proteins, DNA cannot organize into chromosomes, and life as we know it would not exist. Furthermore, histones play a role in gene regulation. As a part of chromatin, histones assist in control of expression, that process by which the coded information in genes is converted into operational structure in the cell. Coming to the next, non-histone proteins. According to the everythingbio.com, a non-histone protein is a protein remaining in chromatin after the histone proteins are removed. This simple statement does not do justice to the important role of non-histone proteins. So the non-histone proteins include scaffold proteins, heterochromatin protein 1. So these are the non-histone proteins, scaffold proteins, heterochromatin protein 1, DNA polymerase and polycom and other motor proteins, all of which play an essential role in the cell structure. In fact, non-histone proteins provide the scaffold structure of DNA and complete numerous other structural and regulatory functions that make life possible. Significance. Histone proteins cannot work alone. Histone proteins can complete their functions only in the presence of non-histone proteins. Yet the histone proteins differ from non-histone proteins in that the histone proteins are highly conserved across the species, whereas non-histone proteins are not. In other words, the histone proteins found in one species are typically found in other species. Both proteins are essential to biology, both are found within the living cells. Both provide a structure to DNA, but always in which they operate differ significantly. Okay, so this is the difference between histone as well as non-histone proteins, right? I think it made somewhat easy the concept about histone and non-histone, DNA, chromatin, nucleosome, and chromosome, right? Okay, if you have any doubts, please post it in the comment box. Thank you very much. Please do like, share, comment, and subscribe to my channel.